produce publication in reputable journals. The academic staff to student ratio is 1/19 and the supporting staff to student ratio is 1/17. To ensure the sustainability of the campus in the future, Universitas Andalas has had an integrated waste management system, an integrated water management system, and a micro hydropower plant inside the campus. 1/19 and the supporting staff to
Ya, halo. Ya sama aja lah. Apa? Boleh yang pilih yang untuk anak kucing satu, untuk induk kucing satu ya, Dek. Semarang aja lah, sama kucing atau apa. Ya.
Well, ladies and gentlemen, may I have your attention, please? Our lecture is about to begin. Thank you very much for preparing yourself to join our guest lecture. Assalamualaikum warahmatullahi wabarakatuh. Good morning all. Honorable Dean and Vice Dean of Public Health Faculty, Universitas Andalas, Excellency, our speakers, Virajali G. Vital, PhD, DPAM. Sorry, Dr. Pai, if I'm wrong to spell your name. Distinguished participants, guests, ladies and gentlemen, it gives me a great pleasure to welcome all of you to the guest lecture about antimicrobial activity and the impact to public health. I'm Prima Elda and Dr. Kamal Kasra, the moderator of today's guest lecture. We are very pleased to see you here in the meeting and welcome all of you to this event. We will have some time for questions and comments at the end of the presentations. As participants, you are encouraged to ask questions, share your thoughts, and present any relevant examples. This is meant to be an interactive discussion, so don't hesitate to contribute with your insights. First of all, let me introduce you to our lecturer, Piranjali G. Vital, PhD DPAM. Dr. Pai is currently university researcher, scientist in the Scientific Career System of the Department of Science and Technology, Civil Service Commissions, and head of the Biological Research and Services Laboratory at the Natural Science Research Institute in the University of the Philippines, Diliman. She is also an affiliate professor at the Graduate School of the University of the Philippines, Los Baños. Dr. Pai has 20 years of experience performing research and teaching in the field of environmental microbiology. She is a prolific researcher with publications and paper presentation, a multi-awarded scientist, as evidenced by several highly prestigious recognitions she received. A generous teacher and mentor, an outstanding woman in science and public servant, and leader being a head of the laboratory and serving several professional organizations. So without further ado, let's get started. And Dr. Pai, time is yours.
Thank you very much, Professor Prima. Good morning, everyone. I am here again to share my screen first. You can see it now, right? Okay, just like make it in full view. Yeah, you can see it. Okay, so the one assigned to me is antimicrobial resistance and the impact to public health. I hope you had a good weekend. So good morning again, everyone. I was already introduced by Professor Freema, so I go straight to... Okay, so I like to define, of course, antimicrobial resistance. What is the impact to public health? And in the second part, I will be sharing with you briefly my research on antimicrobial resistance in the field of agriculture. As mentioned, I am interested into, or I do work on environmental microbiology, and of course, how to be able to prevent them for the third part. So antimicrobial resistance, or some not only AMR but MDR would be multi-drug resistant, is a top 10 global threat to public health, food security, and development, according to the World Health Organization. I know as public health experts, as food safety experts, or as nutritionist experts, you know that this is really a global threat. And as I have mentioned before, like in the past webinars or lectures, I called this as a silent pandemic. We don't see yet the antimicrobial resistance, but in other references, they call it as a super bug. Like they have this AMR. We no longer can use current antibiotics, but we need other types of antibiotics to be able to combat AMR or antimicrobial resistance. When we say antimicrobial, it occurs in microorganisms. So these microorganisms can either be bacteria, virus, fungi, or parasites or protozoa, but I would be concentrating on antibacterial resistance, which is on bacteria. So antibiotic resistance can affect anyone of any age, in any group, in any population. it occurs naturally, but misuse of antibiotics in humans and animals is accelerating the process. And a growing number of infections such as pneumonia. I heard, of course, here in Padang, there's still tuberculosis, gonorrhea, salmonellosis, which is part of food poisoning, are becoming harder to treat, and antibiotics used to treat them become less effective, as mentioned earlier. So antibiotic resistance leads to longer hospital stays, of course, higher costs, higher medical costs, and increased mortality. And what could be the causes of antimicrobial or antibiotic resistance? As I mentioned, I will be focusing on antibiotic resistance, as there is, of course, a lot of microorganisms when you say antimicrobial resistance. So antibiotic resistance happens when bacteria change and become resistant to the antibiotics used to treat the infections that they cause. It can be due to overprescribing of antibiotics, patients not finishing their treatment. Like, for example, we were given 7 days by our doctors, but we just usually like consume it for 3 days because we already feel better, and we no longer like continue the dosage until the 4th day, or simply because we cannot continue anymore because we do not have enough money to buy future or the succeeding days for the antibiotics. Could also be overuse of antibiotics in livestock and fish farming. This is also very alarming because as we are not all working in the livestock or in the farms, so we do not know what antibiotics are being applied to our animals and other livestock. Poor infection control in hospitals and clinics, lack of hygiene and poor sanitation, although of course this was addressed during the COVID-19 pandemic, so we were like super informed of how washing, disinfection could really help us. And then the lack of new antibiotics being developed. That's why others would like go or explore natural products for us to be able to see other antibiotics. And then what would be causing, or what would be the statistics for antibiotic resistance? This is just one of the references. Other references would be saying other percentages, of course, but this is like, why is it a problem? So more than 40 million antibiotic prescriptions from 2011 were unnecessary. I believe some of us would just even go to the drugstore, buy antibiotics, even not prescribed. But now in the Philippines, we need prescription before we can buy antibiotics. I'm not sure in Indonesia, would it be the same? Like you need doctor's prescription before you can buy the antibiotics. Yes. Like before the year 2000, I guess, in the Philippines, we can buy over the counter these antibiotics without any prescriptions. So anyone not feeling well would just buy this antibiotic and take this like vitamins. So no new major antibiotic has been developed in the last 30 years, and nearly 50% of, this is US data, is contaminated with bacteria that is resistant to various antibiotics. And in our case, in the Philippines, and even in Southeast Asia, there are reports of antibiotic resistance at the agricultural level or the sources. So only 10% are prescribed with the correct antibiotic, and nothing for correct prescribing rate for acute bronchitis, of over 4% increase in antibiotic prescription rates in the last 14 years, and only 24% of antibiotics used in the US are for humans. So sometimes we have viral infections, but medical doctors will sometimes also give us antibiotics. So we also know, of course, that if you have virus infection, antibiotics would not be able to cure you because these are not bacteria, but are viruses. So what would be the mechanism of antimicrobial resistance? I'm not discussing this in detail, but this is just an image showing us how antimicrobial resistance would occur or happen. So, of course, in bacteria, there are no organelles. So you just have, of course, your genetic material. You have ribosomes, and of course, in your genetic material, there could be modification, which is a modified drug target. So it can no longer like identify antibiotics either through cell wall, through the genetic material, through the ribosome. You know, of course, that there are different types or classes of antibiotics. While these different classes of antibiotics could target like the ones that I mentioned, cell wall, the genetic material or the ribosomes, they alter their gene, so that it could cause like modified cell wall protein impermeability or inactivation, adding a phosphate group on the antibiotic, which will reduce its ability to bind to the bacterial ribosome, or either pumping out, increasing active efflux of drugs. So antibiotic resistance, they say, even by the World Health Organization, will kill more people than cancer and diabetes combined by 2050. So have you already heard, like, aside in agriculture, if you have like skin infections, like the usual staphylococcus, the MRSA and the VRSA, or MRSA and the VRSA. So I'm not sure if you also have those cases here, but in the Philippines, you have MRSA, and if methicillin resistant is not, then there's also VRSA. So the doctors will not be able to know what type of antibiotic they can they will prescribe. So could be like it could be from the livestock, could be consumed by humans, and could be transferred to different populations. So I go straight to the studies that we have conducted in terms of antimicrobial resistance, as I already gave you a very brief background of antimicrobial resistance, some mechanisms, and of course how it is perceived by the global community. So I have first, I have a recent paper in review on fecal contamination in antimicrobial resistance in the urban gardens in the Philippines. Why such interest? Because during the lockdown, during the COVID-19 pandemic, which is more than a year, the farming in the Philippines translated, particularly in Metro Manila, to urban gardening, because we cannot go out, but we have to make a way to like quote unquote survive. So our local government unit supported these urban gardens, and these urban gardens were also like very supportive. They wanted to see if there's contamination in their food, mainly because they want to see like if their AMR and contamination will be transferred to humans, because the population consume the products that they produce. So of course, antibiotics have been used in different fields in agriculture. It is used on plants and animals to combat diseases. Of course, already mentioned earlier, it promotes AMR of the bacteria in the environment. As antibiotics are completely absorbed, they settle in excrement and are used as fertilizer in the soil. So as mentioned, urban gardening has been like an in thing in the Philippines. So it is a practice of cultivating, processing, and handling of crops and livestock within or around urban areas. Just mainly because I'm from Metro Manila. In Metro Manila, we do not have like enough land, so we stay in condominiums, in buildings, etc. So there's very little land. So those little lands are developed into urban gardens. And with the COVID-19 pandemic, these urban areas in Metro Manila have adopted urban gardening as a source for food supplies. And of course, due to close proximity to urban settlements, it is a hotspot for AMR bacteria. And then these were the methods that we did. We had around 116 samples. We actually had more than like 400 or 500 samples. We just focused on this one. We focused on only 30. And with these samples from vegetables, water, soil, and the fecal samples. I go straight to the CLSI performance standards for AMR testing. So this would be the guidelines for the different antibiotics. I know as public health experts, you are very familiar with the classes of antibiotics. So I will not be discussing them, or I will not be discussing the mechanism. But this would just be the susceptibility, intermediate, and resistant numbers for the zone of inhibition. So this would just be examples of the DDA, or the disk diffusion assay that was done. And for the results, I would go straight to the results. This would be like, as you can see, there would be highest resistance in tetracycline as well as in ampicillin. So just to see it clearly, this would be the resistance percentage per sample. As you can see, almost like 71, 56, and 80% for all the types, as well as 100% for the soil samples for tetracycline resistance. This is followed, I think, by ampicillin resistance. You know, of course, that in tetracycline, the one that is prevented already is the ribosome. So it's very, very difficult to move to another antibiotic if this one is already, if the microorganism is already resistant to tetracycline. And so we just summarize based on that data that we have like the general antibiotic resistance would be tetracycline greater than ampicillin and so on. And then last for ciprofloxacin. Of course, you know that NA and ciprofloxacin are last resort drugs. And it's a good thing that of course is that the microorganisms has very low resistance to these antibiotics. And then as you can see, like the sources, the agricultural sources would really be the source of this AMR as seen in our study, not only in this study, but in also in other papers, in our other papers, that antibiotic resistance would be higher in the water and the soil samples, followed by stool, and the last would be the receiving end, which is the vegetables. So it is a cycle because we consume vegetables and go back again to this sources. So it would just like be running in the community, or AMR would just be circling around the community. So as a conclusion for the first study, urbanization leads to use of the antibiotics, and exclusion may promote more ARB in their community. As I mentioned, it's cycling around. The antibiotics that are both used by urban communities and agricultural setting likely show high antibiotic resistance in urban gardening, and urban gardens still serve as a hotspot for ARB and further aggregate with misuse of antibiotics by the community. So aside from that, we have a second study on antimicrobial resistance. I think I showed this briefly last time, but I want to show it again. Antimicrobial resistance in Escherichia coli and Salmonella isolates from the fresh produce and the impact to food safety. So we obtained like I think hundreds of samples from the markets and the agricultural sources. This was published in the Journal of Environmental Science and Health Part B. So as we can see, there are antibiotic-resistant Salmonella and E. coli isolates from the produce surveyed from open-air markets and the supermarkets. The one that you can see below are the areas or the markets that we went into. So we did not only concentrate in Metro Manila, but we went to the provinces in the north and also in the south part of the Philippines. And as compared with Metro Manila, there was also high antimicrobial resistance in all of those provinces. So, but this data is really very relevant now in our Ministry of Agriculture, particularly in the Bureau of Agriculture and Fisheries Standards, as we are developing or creating Philippine national standards for microbiological criteria in agricultural products and produce. And I happen to be the vice chair of the technical working group. So this data or this publication is not to produce care, but of course to be able to craft Philippine national standards. And as we can see, there's one market that there's really high antibiotic or antimicrobial resistance, as you can see in the graph. This is like the divisory market in the Philippines is like a very big marketplace in the Manila part of Metro Manila. It's more of the southern part of Metro Manila. And so aside from that, we have also observed antibiotic resistance among E. coli isolates from agricultural irrigation waters in central part of Luzon in the Philippines, mainly because the vegetables will not be coming mostly from Metro Manila, but because the sources would be in the provinces. So we wanted to know if the AMR of those 100 samples would be coming from the sources. So we went back to the sources and we sampled different sites. Instead of doing the disk diffusion assay or the antibiotic disk assay, we had molecular techniques to be able to detect AMR. So we detected the class 1 into integrants, as references would say that if bacteria, particularly Escherichia coli, would have class 1 into integrants, it would say that this AMR or antimicrobial resistant or even MDR. So as we can see from this graph, this is just a molecular weight marker. This is the positive control. The negative control should have no bands after PCR and gel electrophoresis. The two bands would signify the presence of the INT1 and INT2 genes, or the class 1 INT2 integrants. And this one would only have the INT2 gene, same for the lanes 12 and 13. So aside from detecting the class 1 into integrants, as well as looking at AMR in the markets, we went back to the sources of fecal contamination, mainly because we wanted to see if this fecal contamination would be directly affecting the community. So we published also this on the Journal of Environmental Science and Health, Part B, particularly phylogroup typing and carbapenem resistance of Escherichia coli from agricultural samples. And we did phylogroup typing because this could be easier. Like we can see easily, as you can see from the abstract, like we can see easily what is the source of the contamination. This is a molecular assay. If you see the phylogroup type like A, B, C, we can already like direct it like this came from this source, etc., or this gene would be coming from the cat, the chicken, the dog, etc. So it would be easy to pinpoint the source of the AMR. So that's the story for this article. So those would just be a few studies that we did for AMR in agricultural sources. And so I go straight to how to address the issue. So we have this, of course, an increasing rate of AMR and even MDR in the community, not only for E. coli, not only for the field of agriculture, but a lot of fields. So in 2015, there was this WHO Global Action Plan on AMR. So the objectives of this is to improve awareness and understanding of antimicrobial resistance, to strengthen surveillance and research, to reduce the incidence of infection, to optimize the use of antimicrobial medicines, and to ensure sustainable investment in countering antimicrobial resistance. Of course, the second one, you know this one, the One Health approach. I will not be discussing that in detail, as I believe I already discussed that in my past seminar or lecture. And then we have what we call also as what we celebrate as the World Health Antimicrobial Week, which is an awareness week for AMR. I'm very sure that also in this country that you have like meetings with the WHO and the other organizations just to address, of course, AMR. And we have this based on the guidelines. You have the Global Antimicrobial Resistance Surveillance System, or the GLASS, Global Antibiotic Research and Development Partnership, or the GARDP, and the Interagency Coordination Group on Antimicrobial Resistance, or IACG. And allow me to just share with you the different prevention and control for different groups. First, for the individual, suggested, of course, we only use antibiotics when prescribed by a certified health professional. We never demand antibiotics if your health worker says you don't need them, of course. Of course, as an expert, you already know that. Always follow your health worker's advice when using antibiotics. Never share or use leftover antibiotics. It's very important for the students here listening in the group. Prevent infections by regularly washing hands. Again, we were already informed of this during the COVID-19. I'm very glad that we have that practice already. Preparing food hygienically, avoiding close contact with sick people, and keeping vaccinations up to date. We also prepare food hygienically following the WHO keys to safer food, like clean, separate, cook, and then chill. Those food that should be kept cool should be cool, should be eaten cool. Food that should be kept warm should be eaten warm. And choose foods that have been produced without the use of antibiotics for growth promotion or disease prevention in healthy animals. And then for prevention and control, suggestion for policymakers: ensure a robust national action plan to tackle antibiotic resistance in place, improve surveillance of antibiotic resistance infections, strengthen policies, programs, and implementation of infection prevention and control measures, regulate and promote the appropriate use and disposal of quality medicines, and make information available on the impact of antibiotic resistance. So prevention and control for the group of health professionals. I know you are part of the health professionals. You prevent infections by ensuring your hands, instruments, and of course, environment clean. Only prescribe and dispense antibiotics when they are needed according to current guidelines. Report antibiotic-resistant infections to surveillance teams. You talk to your patients about how to take antibiotics correctly and antibiotic resistance and the dangers of misuse. And talk to your patients about preventing infections, example, vaccinations, how hand washing, and covering nose and mouth rinses. And of course, prevention and control for the healthcare industry. We invest in research and development of new antibiotics, vaccines, diagnostics, and other tools. Of course, we learned during the COVID-19 pandemic that we should have good diagnostic tools and methods, such as, of course, vaccinations. And then, of course, lastly, in the field of agriculture for prevention and control, for veterinary, here in the group, or vets only give antibiotics to animals under veterinary supervision. Let's not use antibiotics for growth promotion to prevent disease in healthy animals. We have seen this because as we have observed in our data, there was a large AMR in the sources, like from the water, irrigation water, from the soil, and fecal contaminations. You can see that the higher sources would really be coming from the agricultural sources. We only vaccinate animals to reduce the need for antibiotics and use alternatives to antibiotics when available. Promote and apply good practices at all steps of production and processing of foods from animal and plant sources, and improve biosecurity on farms and prevent infections through improved hygiene and animal welfare. So that's all for me. Thank you very much. Terima kasih.
Okay, thank you very much, Dr. Pai, giving informative and interesting presentation. So, dear participants, now we come to question and answer session for about 55 minutes until an hour. We will welcome also participants to discuss about antimicrobial resistance and the impact to public health. Now, I'd like to have 3 questions from the participants, please. Please raise hand or chat, write down on chat column. We invite maybe from Ibu Helmizar. Selamat pagi, Ibu.
Ya, selamat pagi Bu Prima. Thank you very much. Maybe
Please, Ibu, if you have some question to Dr. Pai.
Oh, okay. Thank you very much, Prof. Pai, Dr. Pai. Sorry, I'm not
That's okay.
uploading the video because in the outside. This yeah, I think it is a interesting topic and presentation. Maybe just one question for you for this antimicrobial to apply to public health. Maybe what the consequence on health, and maybe in nutrition, if we use antibiotic or others, that can kill the probiotic in diet rates, maybe. But what your recommendations for applied or implementation for public health or nutrition? Thank you very much.
Okay. Okay, thank you, Ibu. Dr. Pai, we will.
Hi, Professor Helmizar. Hello again. Yeah, you
Yeah.
asked for the consequence in public
Okay.
health, as was already discussed. Of course, consequence in health would be the difficulty of providing antibiotics or antimicrobials to patients. So if they are they contain bacteria which are AMR or even MDR, the health practitioners would have a difficulty in providing prescribing proper antibiotics or antimicrobials to them, and this would impact, of course, the economy. And of course, not everyone of us, I'm not sure in Indonesia, but in the Philippines, not everyone would have the health insurance. So it is not covered. If you're hospitalized, you would be covering it as an individual. So it would be an economic problem for the family that would be staying longer in the hospital. So that's the one that I see in health. And yeah, particularly, for example, like a particular experience in the family.
Yes.
She had like a family member had Staphylococcus aureus infection. So aside from being a MRSA, was also like VRSA. So it took a long time, like a year or more, to cure the family member of the antibiotic resistance. I think that would be the same for you. And then for nutrition, of course, or in terms of food safety, the one that I discussed for the agriculture, that antibiotics are applied in the farms, in the livestock, or even in the farms. It would be very difficult because these AMRs would be traveling or cycling to consumption from the soil, water to animals, the animals that we consume. I think that would be a threat to food safety and even to food security. Connecting that to, of course, the global sustainability problem, which is part of the UN Sustainable Development Goals.
Okay, okay.
you,
Thank you
Dr.
very much for the answer, Prof. Pai. Thank you.
Okay, thank you, Dr. Pai and Ibu Helmiza. Please, if you have any comments or any question to Dr. Pai from our students and our lecture.
Maybe if you have a story like in your university or
Okay.
like in your community, you have a story of antimicrobial resistance, maybe someone would like to share also.
Yeah, maybe if you have some experience about antimicrobial, or antibiotic resistance in our country on or in your project, please share with us. Good morning, Ibu Ade. Maybe Ibu Ade have some experience. Yes. Comparison in Indonesia and Philippines. Okay. Good morning, Pak Syahrial.
Good morning, Ima. Mohon maaf. Ini
Okay, Pak Syahrial. Maybe from our students, you have any questions, Dr. Pai?
Maybe everyone's warming up. It's Monday morning.
Okay. Good morning, Bu Ice. We invite Bu Ice. Maybe Bu Ice have some experience about antibiotic resistance.
Morning, Dr. Pai. Nice to see you again. Thank you for the interesting presentation for today about the antimicrobial biological to the public health. So in our faculty and also our university, sometimes actually I'm not expert on this the study about the antimicrobial activity, but actually I want to study about the PCR lab PCR activity in the antimicrobial. So meaning that if we I saw that the presentation about the molecular weight that you after we did the activity antimicrobial activity, so meaning that we have to analyze the weight molecule or what we should do about the present of the that you presentation previously about the result. So meaning that we have to analyze the weight molecule weight or what about the result and the after we analyze the result the study. Okay, thank you, Dr. Pai.
Hi, Professor. Yeah. For the molecular analysis, it's very easy. It's not like in culture analysis, like the distribution or the broth dilution method. You just need to extract from the samples DNA. As soon as we have the DNA, we can run this in PCR. If you're familiar with COVID-19 RT-PCR technique, I'm sure you have that in the you have RT-PCR in the university, right? It's very similar. It's just PCR. You just put it in the machine. So after 3 to 4 hours, you run it in the gel. So after running it in gel electrophoresis, the bands that I showed you earlier, the lines, those are the bands, would show the gene for antimicrobial resistance because there are primers. The primers are specific to the genes of the bacteria and would be able to amplify them. So in just a half day, you would be able to see already the particular antimicrobial resistance gene. Unlike in this diffusion assay, like you collect your sample today, you swab it in your plate, you incubate tomorrow, you have to check it again the next day, following day, right? But in molecular biology, in molecular diagnostics, it's very easy. It's similar with the COVID-19, like you just swab, do RT-PCR, automatically you can see if it's positive or negative. It's the same. So there.
Okay. It's very, I thought it's very interesting to if we want to do study about the antimicrobial activity. So meaning that if we want to do the food investigation about the impact of food investigation to the muscle mass, for example. So is it okay if we can do with the Western blotting analysis with the antimicrobial of the impact of the food investigation? Which one is better, PCR or Western blotting?
I'm sorry, come again. The second one is?
So if you want to do the study about the muscle mass impact of food analysis for the impact of some disease like diabetes mellitus. So which one is better if you want to do the PCR or Western blotting? Which one is better?
Yeah, for me, I'm biased towards PCR because that's diagnostic. Because in Western blot, from the term itself, blot, you have to like transfer, and that will take another time also. It's another technique though, if you want to see like a particular gene or protein in your sample. But since you just only want to detect, PCR would be easier. PCR plus gel because that's qualitative. But if it's quantitative, you have more fun, you can directly go to RT-PCR like COVID-19. Detecting COVID-19, it's easier.
I thought it's very interesting idea in order to conduct the study in the future. I will tell about the PCR and antimicrobial activity.
Yeah, we can. And our lab also has like workshops. I'm not sure if we will have like face-to-face or hybrid or online for this year, but we have molecular biology workshops. Maybe you can visit us.
Okay. So for example, like this one, in antimicrobial, and also like um, anti in the yogurt, for example, in the yogurt, if we put some substitute sorry, I mean the fortification in the product like in the bread. So how can we make sure that the microbial is still available in the product after we did the food analysis?
Yeah, I think that's the next question, but it's not related to antimicrobial resistance. You just want to see the population. If easy, you want to see like quickly, you can do NGS, next generation sequencing. You can like extract community DNA and then have it sequenced. With this NGS, you can see the entire population present in your sample. We did that already, and we were successful in amplifying the bacteria present in the probiotics. You can do that. If not, you can go traditional methods, like you can still do PCR and gel electrophoresis, but you can use 16S rRNA sequencing.
Okay. It's very interesting. So maybe we can do the conduct study about in the future. So because in our department we have three major about the concentration like clinical nutrition, public health nutrition and also food food
Safety.
analyzer. So in the antimicrobial like yogurt in our food
Yogurt.
analysis. I just
Yes.
yogurt, yeah. I just want to make sure if we did a food fortification, how we can make sure that we still have the benefit of the product in our food analysis after we finish food processing. Thank you. Thank you, Prof. Pai.
Thank you also, Prof.
Okay, thank you, Bu Ice and Dr. Pai. Thank you. We also invite our students to give some questions to Dr. Pai. Is it okay if you ask in Indonesian and we will translate to Dr. Pai? Silakan adik-adik atau teman-teman yang ingin bertanya, tidak apa-apa dalam bahasa Indonesia, kita akan bantu nanti untuk translate dalam bahasa Inggris ke Dr. Pai. Jadi tadi Dr. Pai menjelaskan bahwa I will translate in Indonesian to our participants. Jadi Dr. Pai tadi menjelaskan tentang bagaimana dampak antimikrobial dan antibiotik ini resistennya, yang dimana antibiotik resisten ini menjadi salah satu isu atau global threat to public health. Jadi dalam isu public health, dalam food security, kemudian dalam mikroorganisme lainnya. Jadi ini menarik sekali ketika kita bahas. Jadi adik-adik yang tertarik atau mempunyai pengalaman atau dosen-dosen yang mempunyai pengalaman terkait dengan riset pada antimikrobial atau antibiotik ini mungkin bisa di-share pada sesi kali ini. Silakan adik-adik.
Potensi di putaran pertama itu baik Pak.
Please, our students or our lecturers, if you have any question or experience to share with us. We invite good morning, Ibu Nila Kusuma. Oh, very. Okay. Sorry. Oh, okay. If we have no more question, we will. Sorry. We will invite Bu Ade first. Maybe Bu Ade want to ask you something. Oh yeah, Bu Ade. Do you have any comments or question to Dr. Pai? Ibu Mary? Pak Sahrial? No more question, Pak?
No question.
Okay. Ladies and gentlemen, if you have no question, we finally come to the end part of this guest lecture, antimicrobial resistance and the impact to public health. Before we close this session, I would like to invite Dr. Pai to give us some closing remarks. So Dr. Pai, please.
So thank you, Professor Frima, for moderating this session. Thank you, everyone, for listening. This is my last day in Universitas Andalas. If you want to continue, of course, to communicate with me, please do so. I am hearing good probable collaborations and possible connections for research in terms of antimicrobial resistance. So thank you and have a great week.
Okay, thank you for the concise closing remarks. Please allow me also to take some highlighted points from this guest lecture. The conclusion that urbanization leads to excessive use and excretion of antibiotics and may promote more ARB in the community. And antimicrobial resistance, or MDR, is top 10 global threat to public health, food security development, occurs in microorganisms such as bacteria, virus, and fungi. Antibiotic resistance happens when bacteria change and because resistant to the antibiotics used to treat the infection they cause. So for the prevention and control in individual, only use antibiotics when prescribed by a certified health professional. Jadi jangan pakai antibiotik begitu saja tanpa ada resep dokter, ya. And never demand antibiotic if your health worker says you don't need them, and always follow your health worker's advice when using antibiotics. Never share to use leftover antibiotics. Prevent infections by regularly washing hands, preparing food hygienically, avoiding close contact with sick people, and keeping vaccination up to date. And last, prepare food hygienically, following the WHO keys to safer food, and choose foods to have that have been produced without the use of antibiotic for growth promotion and disease prevention in healthy animals. So Dr. Pai, thank you very much for your insightful presentation, and thank you for your attention, dear participants. We have come to the end of our guest lecture. I hope you enjoy. I hope you have enjoyed this session and gained some valuable insight and knowledge from our lecture. Hopefully, this guest lecture will be beneficial for everybody. Amin. Please give applause for the speakers and for your. Okay, thank you very much. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Okay, thank you very
Jangan
much,
Dr.
Pai.
Pai.
Nice to meet you
Dr.
again.
Pai.
Dr.
Okay.
Pai, thank you very much.
Jangan lupa untuk bisa absen ya Bapak Ibu sekalian. Absensi ada di kolom chat dan saya akan present kembali.
Belum ada Bu Ima.
Iya, ya, saya akan share kembali.
Sudah 1 minggu Mr. Pai ini.
Oke. Dr. Pai, hopefully you enjoy in Padang.
Oke, izin live. Oke, thank you.
Iya. Oke, saya sudah share link absensi Bapak Ibu sekalian. Bisa
Makasih
untuk
Bu Ima.
ya, terima kasih semuanya. Dr. Pai, thank you very much.
Yang jam 11 masih itu Bu Ima.
Sebentar Pak, mau dikonfirm dulu ya Pak. Sebentar ya Pak, untuk yang jam 11.
Kan nggak ada bawa apa-apa ya?
Enggak ada Pak, cuman untuk diskusi aja.
Iya.
Oke. Oke.